Assalamu alaikum and good morning. In the series of virus lectures, this is the fifth lecture, which we are going to talk about human viruses that are involved in cancer. Now, the main objective here is that you should be able to list the agents that are associated with cancer, the microbial agents, list both tumor-causing DNA and RNA viruses, and discuss what is the evidence that makes those viruses associated with the cancer, and what sort of preventive measurements could be used to prevent some viruses from causing cancer. Now, cancer and its causes. What is cancer? Cancer is a rapid creation of abnormal cells that grow beyond their usual boundaries and which can then invade adjoining parts of the body and spread to other organs. The process of transformation of normal cells into tumor cells in a multi-stage process that generally progress from a precancerous lesion to a malignant tumor. So that is the process which involves the formation of cancer. Cancer is known to cause around 9.6 million human deaths globally. That's according to the statistics of 2018. And about 1 in 6 deaths is due to cancer. Nearly 70% of the deaths from cancer occur in low and middle income countries. Cancers are caused by abnormalities in the genetic material that causes carcinogens, example, tobacco smoke, radiation, chemicals, or infectious agents. Viruses are the second most important risk factor for cancer development in humans, and cancer-causing infections such as hepatitis and human papilloma virus are responsible for up to 25% of cancer cases in low and middle income countries. Oldbank and Wilhelm Ellerman introduced the theory that cancer could be caused by a virus. This was confirmed for solid tumors in chicken in 1911 by Peyton Rouse. Now, the viruses that are associated with human cancer are as follows. We have human papilloma, hepatitis B, Epstein-Barr, human herpes type 8, and human T-cell leukemia virus. We'll be talking about each one of them individually as we go on. Of course, not only viruses can cause cancer, there is also the bacteria. So we shouldn't forget bacteria, for example, Helicobacter pylori, which causes gastric lymphoma and adenocarcinoma, which are detected in majority of cases of gastric lymphomas. Antibiotic treatment results in regression in 70% of those cases. Campylobacter jejuni, intestinal lymphomas. Other types of bacteria can also cause cancer. But mainly here we are going to talk about viruses, since they are well known and have been studied very widely in this aspect. How do viruses cause cancer? Usually there are different ways or mechanisms that lead to the formation of cancer. Either through acute transformation, usually the virus has a gene called viral oncogene, and the expression of this gene causes overactivity of the host oncogenes, and therefore they cause transformation of the cell. And then we have the other way or mechanism, which is the slow transformation, where the virus genome is inserted near a proto-oncogene. And the proto-oncogene is present in the host genome, Therefore, it causes overexpression of the proto-oncogene, which in turn induces uncontrolled cellular proliferation. Now, for human papillomavirus, this virus is well known to cause genital cancer. It plays a role in a variety of cancers. It's associated with skin cancer and cervical cancer. In 1978, Jablonska and Gerard Orth at the Pasteur Institute discovered HPV-5 in skin cancer. So human papillomavirus type 5. In 1983 and 1984, Zorhausen and his collaborators identified human papillomavirus type 16 and 18 in cervical cancer. Of course, Zorhausen, for his discovery and his role in identifying papillomaviruses as the causative of cervical cancer, he received the Nobel Prize in Medicine in 2008. Now, these viruses or human papillomaviruses have something like 100 types. They cause a variety of hyperproliferative lesions in stratified squamous epithelium at various anatomical sites. These viruses can be divided into two groups based on the site of infection. So those infecting the cutaneous site, example, the commonly HPV types 1, 2, 4, or those infecting mucosal surfaces, example, the commonly HPV types such as 6, 11, 16, and 18. Note why that the viruses are typed on the similarity of the genome and not serologically, meaning that those viruses vary from each other in the divergence of the sequence, how much the sequence are similar and how much they are far from each other. Most of the lesions are benign and regress spontaneously. However, the mucosal viruses are associated with pre-malignant and malignant diseases of the oral and genital tract. The commonest cancer associated with HPV is cervical cancer. We know that HPV 6 and 16 are the common cause of genital warts worldwide. The warts are benign and rarely become malignant. Therefore, HPV 6 and 11 causes benign disease, while 16 and 18, or human papillomavirus 16 and 18, are the commonest cause of pre- and malignant disease of the genital tract worldwide. They are found in cervical and penile lesions. How is this achieved? What is the mechanism behind the virus being able to transform the cell? Usually the virus has two oncogenes. We're talking about human papillomavirus, the E6 and E7 gene, or early gene 6 and early gene 7. They modify the cell cycle in a state that is favorable to the amplification of viral genome replication and consequent late gene expression. Usually E6, early protein 6, causes degradation of p53, while E7 competes for retinoblastoma protein, or PRB, binding, therefore freeing the transcription factor E2F to push the cell cycle forwards. So clearly, from degrading the p53, which is a suppressor protein for the growth of the cell, once this is degraded, then the cell is going to be able to grow uncontrollably due to that situation. So in the mechanism of pathogenesis of HPV, there are different stages. It all depends on what sort of progression is the virus going to go through. Either it's going to go through the latent infection, or it's going to go through the productive infection. And again, the metaplastic epithelium infection either is going to lead to the integration of HBV in a later stage with the DNA of the or chromosomal DNA, and the production of early 6, early 7 proteins, therefore inducing the cells to become malignant, or the immune response is going to lead to the regression of the infection and the disappearance later on of the virus. Now, again, there are, not to forget that there are cofactors that might induce this process, the process of transformation, such as sexually transmitted infections, smoking. All these can help in the progression to the cells, for the cells to become malignant cells. What is the proof of association with cancer? Usually what we tend to see is that the viral DNA is found in 95% of pre- and malignant tumor cells. Viral DNA. And usually infection precedes the disease. So before you get the tumor, usually we tend to, or cancer, we tend to see that the person has been infected by this virus. And we know that animal papillomavirus have been shown to cause tumors in natural infections and experimentally. HBV 16 and 18 cells in vitro also are affected, while HBV 6 and 11 are not affected or not transformed. Not everyone infected with these types will develop pre-malignant disease, and even fewer will go on to the malignant stage. Additional factors apart from infection involved in the disease development and progression are unknown. However, 95% of people with either cervical or penile cancer have the viral genome detectable in the cancer cells. Transmission, we know that sexual contact is the most common form of transmission. Both group of viruses, whether it's HPV 6, 11, or 16, 18, can be detected in individuals without disease. Highest incidence is in sexually active people who are among the age group of 18 to 30. In pre-malignant cells, the viral DNA is separate from the chromosomal DNA and is replicating and producing infectious virus, while in most malignant cells, the viral DNA is stably integrated into the chromosomal DNA, resulting in no viral particle production. Now, the issue here is it is not clear if the integration is a prerequisite for conversion to a malignant phenotype. Almost all cervical cancers are related to human papillomavirus. And in 2006, a vaccine was approved that protects against human papilloma type 16 and 18, which together cause 70% of cervical cancer and 90% of genital warts. Recently, in the States, FDA approved a 9-valent vaccine. And this one can protect against 9 different HBV types. The second virus, which is also involved in causing cancer, is hepatitis B virus, which leads to their infection to hepatocellular carcinoma in certain stages. In US, we know that 95% of hepatitis B cases are cleared after acute infection, while 5% of the virus will persist and the individuals will become carriers. That means that they can transfer the virus to other people. There is an association between the rate of the carrier state and hepatocellular carcinoma. In countries where the carriage rate is high, such as around 10% in China and many parts of Far East, then hepatocellular carcinoma is seen to be very common. An additional factor is that in these countries, the virus is transmitted to the neonates at birth or early in life through close contact with the mother. So transmission of hepatitis B routes of transmission include blood, milk, and possibly saliva. Prospective studies have shown that hepatitis B virus positive persons have a 200-fold high risk of developing liver cancer than hepatitis B negative persons. Vaccination and hyperimmune anti-HBV globulin is recommended for neonates born to mothers who are carriers or who have hepatitis B, who have been infected with hepatitis B. What is the proof of association with cancer? First of all, there is a long incubation period before the development of hepatocellular cancer, usually 4 to 5 or even 5 decades. That means 50 years. Note also that relatively few infected persons will develop the tumor. The mechanism by which hepatitis causes the cancer is unknown, but the persistence of the virus with hepatocyte usually damage and produce inflammatory reaction, which may in turn lead to rounds of repair and hyperproliferative response. So this continuous damage and repair may stimulate expression of proto-oncogenes. This is how it's thought to be. Primary liver cancer is the third leading cause of death. Hepatitis B and C are leading causes of liver cancer. And according to WHO, at least 550 people die each year from primary liver cancer. Survival rate for liver cancer is still below 10%. The third type of viruses which causes human cancer is Epstein-Barr virus. Now, we know that Epstein-Barr virus was the first human virus to be directly implicated in carcinogenesis. It infects over 90% of the world's population. So EBV is present in large amounts or infects large amounts of the population. And also EBV has been implicated in the pathogenesis of Burkitt's lymphoma, Hodgkin's disease, non-Hodgkin's lymphoma, nasopharyngeal carcinoma and lymphomas, and epithelial malignancies such as breast cancer. What is the mechanism of transformation in this situation? We know that the latent membrane protein, LMP1, usually mimics an activated growth factor receptor. Mimics means it try to be similar to. So LMP1 is able to transform B lymphocytes in this situation, while the nuclear antigen or IBNA play a role in immortalizing of B cells, converting them into permanently growing lymphoblastocell cell lines. So those two proteins which belong to the virus usually are those which are involved in this process. What is the proof of association with cancer? Again, the viral genome is found in monoclonal integration meaning that the tumor mass developed, which started from a single parental cell, contains the EBV genome. Usually EBV can immortalize B cells in vitro. So from experimentation, we have found that EBV can cause those cells to become cancer cells. Two types of cancer which are geographically confined can be seen with Epstein-Barr virus. The first one is Burkitt's lymphoma, and usually this is found exclusively in the central of Africa. It is one of the most common childhood cancers in the region. This region of Africa has a high incidence of malaria, and this parasitic infection usually is thought to play a role in the development of lymphoma by allowing the increased replication of virally infected B cells. So both the presence of Burkitt's EBV and malaria tends to help in the generation of Burkitt's lymphoma. And therefore, this is going to lead to oncogene activation, c-Myc, and tumor development. Thus, EBV acts to initiate a multi-step, multifactorial process that can result in the emergence of tumor. What is the proof? The lag between virus infection and tumor development for classical Burkitt's lymphoma in African children and for immunoblastic lymphomas in persons with AIDS or transplant recipients is unusually brief. So this lag between the infection and tumor development. Chromosomal translocation resulting in the activation of cellular proto-oncogenes are very common event in lymphoid tumors, not only those caused by EBV. So in Burkitt's lymphoma, the most frequent translocation involves the c-Myc from chromosome 8 to the immunoglobulin heavy chain locus on chromosome 14. So therefore, the chromosomal translocation plays a role again in inducing the proto-oncogenes to be activated. The second EBV-associated cancer is nasopharyngeal carcinoma, which is found in parts of People's Republic of China, especially around the coastal provinces. The cofactor which confines this disease to China is not known till today. But most of the cases or the nasopharyngeal carcinoma cases usually occur in the cities which are near to the shore or the sea, whether it is due to certain habits or of eating seafood or what is it involved, it's not clear till today. Recently, EBV has also been associated with lymphoproliferative disorders, including lymphoma in immunodeficient hosts. Transmission through respiratory routes as seen in natural infection elsewhere. What is the association with cancer? Viral genome is found in cancer cells, very often as a monoclonal integration, meaning that the tumor must develop from a single prenatal cell that contains the EBV genome. Antibody levels of EBV are quite high, and we know that EBV can immortalize B cells in vitro. So all these are factors that associate EBV with cancer. The fourth type of viruses is human herpes virus type 8, and human herpes virus type 8 is associated with different types of cancer such as Kaposi sarcoma, primary effusion lymphomas, multicentric Castleman lymphoma or B-cell lymphomas, and B-cell in saliva may transmit the infection. Usually detection in semen is controversial. Now, rate in general population varies geographically and mirror the prevalence of Kaposi sarcoma. What we see here is that North America and North Europe has the lowest seroprevalence with little to no Kaposi sarcoma. In the Mediterranean and Eastern Europe, including Caribbean countries, intermediate seroprevalence and Kaposi sarcoma incidence are present. While in Central and South Africa, the highest human herpes virus type 8 seroprevalence and Kaposi incidence in the world has been found. What are the risk factors affecting the prevalence of human herpes type 8? First of all, it's considered to be a necessary factor for Kaposi sarcoma to develop. However, other different risk factors can contribute to the prevalence of human herpes type 8. For example, ethnicity, age groups, patients' immune status, or whether they are immune suppressed or not, human behavior, genetic susceptibility, and possible environmental risk factors. So all these different factors can play a role or play a multifactorial role in the development of Kaposi sarcoma. Human herpes type 8 is the most recently identified human oncogenic herpes virus. It was first found by Yan Chang and Patrick S. Moore in 1994 through isolating DNA fragments of herpes virus from a Kaposi sarcoma tumor in AIDS patient. Now, what is Kaposi sarcoma? Usually, it's a rare vascular tumor. It's described initially in elderly men of Mediterranean or Jewish descent, and this is the classical Kaposi sarcoma. Prior to AIDS epidemic, Kaposi sarcoma was also more in East and Central Africa, African endemic Kaposi sarcoma. Increased incidences was also reported in immunosuppressed organ transplant recipients. AIDS epidemic changed the epidemiology of Kaposi sarcoma, and it was associated with HIV-infected patients, meaning the epidemic or AIDS-associated Kaposi sarcoma. What is the mechanism behind the development of Kaposi sarcoma? Human herpes type 8 encodes a viral protein which is referred to as interleukin 6. In contrast to the human interleukin 6, the viral interleukin usually binds directly and activates the shared human cytokine signaling receptor GP130 without the requirement for pre-complications to a specific alpha receptor. Now, the last and final type of viruses is human T-cell leukemia virus, which is involved in T-cell. So what is the mechanism here in human T lymphotropic virus? Usually the interaction between HTLV1 and cellular immune system can be divided into two ways. Either the viral interference with functions of infected host cells, or the subsequent interaction between the infected T cell and the cellular immune system. Usually, human T lymphotropic virus induces transformation by viral protein tax. This tax has the ability to influence transcription activation, signal transduction pathway, and cell cycle control, in addition to apoptosis. So this group of functions usually are controlled by or influenced by the TAX protein, the viral TAX protein. So regarding the pathogenesis of human T lymphotrophic virus, the recent evidence suggests that TAX may promote the G1 to S phase transition, although this may involve additional proteins. A role for other viral proteins, HBZ, that may activate the interleukin-2 receptor pathway has also been suggested in certain cases. HTLV1 infected T cells can non-specifically activate resting uninfected T cell through virus-mediated upregulation of adhesion. Human T cell leukemia virus causes T cell lymphomas. Endemic infections and cancer are restricted mainly to Japan and certain areas of Africa. However, the virus is spreading into other countries and nations such as US, and mainly among the intravenous drug abusers. Now, what is the proof of association between HTLV and cancer is the seroepidemiology survey associated virus with acute T-cell lymphomas and their close contacts. So this is geographical clustering of high incidence by comparing the seroepidemiological results with the development of T-cell lymphoma. And infection of lymphocytes in vitro results in immortalizing T cells. Also, HTLV1 has been shown to be oncogenic in certain animal models such as rabbits. How this virus is transmitted? Usually it's transmitted by infected T cells only. The virus is highly cell associated and free virus in blood is not detected. So now we spoke about these 5 examples of different viruses, their association with cancer, and the proof of how they are associated and their mechanisms to a certain degree. If you feel you have any questions, please do not hesitate to contact me on my email, and I will try to reply as soon as possible. Thank you very much for listening.